My name is Marco Davila. I'm the Senior Vice President of Translational Research, and I'm also the Service Chief for our CAR T-cell therapy service. There's been a lot of success using gene-engineered cells for patients with hematologic malignancies. The best example of this is the CAR T-cell therapy, CAR standing for chimeric antigen receptors. So there are approvals for patients with multiple types of B-cell lymphomas, as well as multiple myeloma. However, the major problem that the community has had is translating these successes from hematologic malignancies to solid tumor malignancies. Solid tumors are more resistant to immune therapies than hematologic malignancies. And it's our hypothesis that this is due to the tumor microenvironment. The next generation of CAR T cells that we are developing for solid tumors rely on armors. And the idea behind this is we add in other genetic engineering technology to make the CAR T cells more resistant to the hostile tumor microenvironment. When we started developing a solid tumor CAR for hepatoma, we wanted to target the antigen GPC3 because this has been investigated in other clinical trials and is relatively a safe target. But what we wanted to armor these GPC3 CARs with is an ability to be able to resist the hostile hepatoma tumor microenvironment. So for this, we're actually doing CRISPR genome editing. So we're editing out the receptor or the gene for TGF-beta R2. So this is a receptor for the transforming growth factor beta cytokine, which is a highly immune suppressive cytokine. So our hope is that when we edit this receptor out of these CAR T cells, the GPC3 CAR T cells, and put them into patients, that it'll be able to not be suppressed by this suppressive cytokine. So we've done initial in vitro and in vivo studies to show that these CAR T cells work and they are able to resist TGF-beta suppression. Now that we've done the preclinical modeling demonstrating the potency and safety of this, we also had to kind of show that we can make these cells in a GMP, good manufacturing practice way. So we've recently expanded our facility from a 6-room facility to a 20-room facility, which is the largest GMP cell and gene therapy facility in the United States in an academic medical center. Every patient that will be enrolled on this clinical trial will have their cells collected and engineered with the CAR as well as the CRISPR edit right here at Roswell Park. So now I'm happy to invite my colleague, Dr. Krishnamurthy, to discuss this first-in-human clinical trial for patients with hepatocellular carcinoma that's available only at Roswell Park and is currently open and actively recruiting patients.
Hi, my name is Anuradha Krishnamurthy. I'm a GI medical oncologist, and I'm also the Associate Director of Early Phase Clinical Trials in Solid Tumor Cell Therapies at Roswell Park. This anti-GPC3 CAR T-cell therapy targets GPC3, a type of protein that's expressed in 77% of hepatocellular carcinoma, but is rarely expressed in normal tissue. The patients who are eligible for this trial are patients with advanced, unresectable, or metastatic hepatocellular carcinoma who have previously progressed on or have been intolerant to checkpoint inhibitors and tyrosine kinase inhibitors such as lenvatinib. Patients should have good performance status and should have at least Child-Pugh Class A liver function. This CAR T-cell therapy is not any different from other CAR T-cell therapies that we administer for hematologic malignancies. The requirement of these trials are that we bring patients in, make sure they are eligible candidates for the trial, and then we get T-cells from them. Then we will manufacture them, and then we will administer the CAR T-cells to the patient. Patients typically need to be admitted to the hospital, and then we will monitor them to see how well they're doing. Patients who are progressing really well with very few side effects may end up being discharged from hospital relatively quickly. Roswell Park is very well equipped to handle any complications and side effects that may arise from CAR T-cell therapy. Advanced hepatocellular carcinoma is not curable. and is a very difficult disease. The testing can be done here at Roswell Park. The recommendation is that you send appropriate candidates for this trial to us, and we will do the GPC3 testing here to see if they meet that final criteria of GPC3 expression. And if they do, we will work with you to provide your patients with the care that they need.